Good.
Thank you, Anno. Okay, today our colloquium series opens, I would say, a new avenue. We have the first talk on environmental science. It's my pleasure to introduce Jeff Warren from Oak Ridge National Park, where he is a senior staff scientist. He is also an adjunct associate professor at University of Tennessee, Knoxville. And he will give a talk with a very teasing title, Climate Change, Roots and Neutrons. Is there a link? Please, Jeff, the floor is yours.
Excellent. Thank you, Kim and Sindra and the organizing committee for inviting me to this symposium. I'm really excited to share my work with you guys and get some feedback and think about how we can move forward in the future with application of neutrons to problems related to environmental science. I've got a presentation today. I'll try to get through it. I'm going to set my timer just so I don't go too long. I'll share my screen. Are you able to see the screen?
Yep.
Excellent. The outline to my talk here is that I'll talk a little bit about our climate change program at Oak Ridge National Lab. The ecosystems that we're working in and the modeling that we're doing, why neutron imaging can be used to help us with our science. The neutron sources at ORNL, I'm not really going to go into those much. I understand you'll have another talk coming up next month about spallation neutron source. And then I'll show some examples of the neutron imaging of plants and soils and looking at structure. Dynamics is really one of the key things that we're interested in at our scale with living organisms and dynamic moving water moving through soils and water uptake by plants and interactions between roots and soils. Water is obviously a big component of our work. Some of the analysis and modeling we're doing and future directions. Oak Ridge National Lab in Tennessee, we've got a Climate Change Science Institute that's brought together multiple different groups to work together to address climate change, specifically focused on improving Earth system models. These are the types of models that are used by the IPCC to project future climate scenarios. And so we've got a group of data scientists, a group of modelers, and a group of empiricists like me. And so most of my work is going out into the forest and measuring ecosystems under environmental stress. One of the things we're really focused on is trying to understand where we're headed in the future for climate change. And looking back in the past, we have had some strong deviations from current conditions, but this has been the very, very far past. The more recent past, over the last 10,000 years or so, we've been in fairly nice Goldilocks conditions here, with not a lot of changes since coming out of the last glacial minimum. But currently we're really on trajectory to shoot straight up this the end of this curve here with really strong increases in temperature. looking back through time, we've got atmospheric carbon dioxide has been fairly constant, between 200,000 and 300,000 ppms over time. And plants have really adapted to 350 parts per million or so. And in the recent years, we've really shot up dramatically. And now we're up over 415, 420 parts per million. This is sort of showing the trajectory of where we're headed based on energy use and societal decisions. Some of the new work coming out of the AR6 Intergovernmental Panel on Climate Change is giving us different scenarios for future conditions based on carbon dioxide emissions. And so on the x-axis here, we see carbon dioxide emissions that are increasing since 1850 here, basically, and showing a pretty linear increase as we're going into the future. And there's a direct correlation here with temperature. And this is global temperature. And if we broke this down into regional temperatures, we'd see more northern latitudes. Northern Sweden would be much, much more affected with temperatures of these higher scenarios reaching 5 or 6 degrees C. And these are different scenarios here that we're tracking out. You know, if we quit using oil and coal, we may end up in this type of blue zone here. If we use all the oil and coal we can get our hands on, we'll be up here. One of the big things to understand here is that there's a wide range of variability around these scenarios. And so it may be anywhere from 1.5 to 2.5 or even 3 degrees C range where we're just not sure. We're uncertain about where we're going to be. And that's where improving the models comes in. And these different models they use for the IPCC reports, there's several dozen different global Earth system models. And the Earth system models have 3 components. They have a land component, they have an oceanic component, and they have an atmospheric component. The main focus for our group has been the land component. And of that Earth system model, of that land component, there's about 100,000 lines of code that are written to recreate these 3 different sort of parts of the land component of the Earth system models: the energy balance, the energy fluxes, the amount of sunlight and radiation coming in, being emitted, the hydrology, the precipitation, the transpiration, and the water cycle, and then the biogeochemical cycles, things like photosynthesis, carbon allocation to different parts of plants below ground, nutrient uptake, nitrogen cycling as well. What we know is that we've got very good understanding when we look at the models. We've got very good understanding of something like photosynthesis and its relationship to vapor pressure deficit, to drought, to water availability, to CO2 concentrations, to sunlight. And we know how leaves will take up carbon, how much carbon they'll take up, how much they'll respire under a wide range of conditions. What's much less certain is what's going on below ground, because we can't just measure it. And so oftentimes we treat this below ground as sort of a black box. And that's where we really want to get into neutron imaging. This root system is sort of essentially the same type of thing as the leaves, and the leaves are out in the atmosphere, interacting with the atmosphere around them and environmental conditions around them. Similarly, the fine roots, or the active parts of the root systems, are below ground, and they're interacting with the bulk soil. And they create this unique environment around them called the rhizosphere that's got a unique biological, chemical, hydrological, and physical sort of structure that's different from the bulk soil. And there's all these different exchanges of nutrients and water and carbon between the root and the rhizosphere in different directions at different times under different conditions. And this is you know where the science is really ripe for new understanding and new representation of these processes in these Earth system models. Here's sort of what some of the models have looked like that we've worked with. And they'll have a canopy component, they'll have a stem component and a soil component. In this case, G'day doesn't have any roots. In this case, they've got 2 blocks of roots, and these are just basically sort of carbon sinks, and there's no interaction with the soil around them. CLM is getting more roots with more of a realistic distribution, but again, not much function applied to these roots. And that function that is applied doesn't really have a dynamic, it's not able to change under different environmental conditions. So we know that the soil resource extraction, plant growth, plant health is linked to root morphology and distribution.
Out of the fence.
Yes.
You're weird.
Yes.
What do you have there?
So I guess I
And,
wrote on and then the pens went down and then I left it.
There's some dynamic root functional response.
This is a meeting. I don't know why, but I was.
Somebody is not muted. Thanks.
Jeff, now you are muted.
Sorry, Jeff, your microphone is off since maybe a minute.
There we go. Can you hear me now?
Yes, perfect.
Excellent, thanks. So we know that these roots are important. We know that they have a lot of interaction and dynamic functionality. We know that if we can include some of this root functionality within the models, we should improve our model estimates of carbon uptake and reduce the uncertainty in these Earth system models that are used to project climate change. And so that's where the neutron imaging comes in. Our work has largely been at the high flux isotope reactor at Oak Ridge. And this is a reactor-based source as opposed to the spallation source. And this will actually be going offline in a couple of years for retrograde for improvements. Here's the cold beam hall here, and they're basically going to be extending this out and giving us much more range of capabilities. I think most of you understand why neutrons are useful and important for biological systems. And it's because of the strong attenuation of a neutron beamline by hydrogen. Whereas some of the larger and heavier elements have smaller cross sections of neutron cross sections. And so hydrogen obviously is strong, key component to water, key component to biological samples, plants, leaves, roots, woods, and soil organic matter is another big component here. We can use neutrons to measure across different scales. A lot of these scales that are focused on looking at structure, high resolution structure, which is based on indirect sort of statistical measurements of the interaction of specific elements and atoms with the neutrons. The work that I'm doing is sort of branching between this neutron microscopy and neutron imaging. And what we're really interested in is being able to see a large enough part of the plant system to understand the interaction of that system with the surrounding media. At the same time, we're interested in being able to see very smaller structures and smaller interactions between soil particles and roots per se. And so really we're sort of spanning this, micron, submicron to centimeter, ideally meter range. Here's the imaging beamline at Hypha that we're currently using, and the flight tube is up here. The neutrons are produced by a reactor in the background here, and neutrons accelerate down this helium-filled flight tube and interact with objects that are placed on the stage in front of this detector system. This is basically what we're seeing when we put a plant inside in front of the detector system. We can put different size plants. The detectors we're currently using are only about 7 centimeters in on a side. And so we can't see something like this entire chamber filled with a grapevine here. And in order to image this, we need to take multiple images. But we can then put in a larger plant with a larger root system and image smaller parts of it. We can also manipulate the environment in the beamline. In this case, we've added light to stimulate water uptake and leaf growth, leaf carbon uptake. Here's an image of a number of different radiographs that were collected across two different of these two different chambers, growing young poplar trees, seedlings, and this grapevine. And so these 2D images are able to scan down then and be able to see the entire root systems. We can splice these then together. Here's another plant system we've got in the beamline showing switchgrass, which is a bioenergy crop that we're interested in understanding its root structure and function under different conditions, and maize, corn. And what we see is really exciting when we look at these systems with neutrons. Not only can we see the root systems very strongly, even the finer roots under fairly dry conditions. One of the most exciting things we saw here and that we're really interested in pursuing additional research on is this dark mass over here associated with the switchgrass. And this is fungal mycelium. These are symbiotic organisms, fungi that associate with the roots of the plant, and they have much smaller diameter hyphae than the roots, which enables them to interact with the rhizosphere and the bulk soil and get into smaller pore spaces, accessing nutrients, exchanging carbon with soil surfaces. And so really understanding this interface between the switchgrass roots and the mycorrhizae, these hyphae, and the bulk soil is really a focus of interest now for us. We can also see the distribution of water content basically in the soil. Here, as we filled this container, we were pouring in soil that was dry, and it was separating out by particle size. And that in turn led to differential water retention by these different size classes of particles. So another interesting thing that we can see and use to understand soil water retention, soil water release under different conditions. Here's this rhizosphere, and this is the really cool sort of place to be. This is actually not the rhizosphere itself, but this is a micrograph, a microscopy slide of a root showing this 200 micron diameter switchgrass root. You can just see part of it here. It's got these root hairs that are sort of a 12 micron diameter range that extend out. And if there were soil here, it would be extending out, creating a bridge and a pathway for movement and exchange of particles of nutrients, of carbon, of water to the root. And then here's this hyphae. And these can go out meters into the soil. So really understanding this linkage and how this linkage may change under dynamic conditions, under drying conditions. As soils dry, in some cases, they shrink. And as they shrink, can we maintain a pathway of connectivity between the root and the soil? And there's potential that hyphae could do that, or do the hyphae break? It's something we're interested in looking at and understanding. Root growth is something that's easily seen with sequential radiographs. And here's just showing a couple of different radiographs showing these root tips growing. Here we've added some water, a pulse of water to the system, and the roots are still growing. We've got some differential retention of water in this zone due to a sand layer that we'd set up in this zone here. And this actually, you can also see that many of the roots are growing in this zone. And then the roots grow on out of the picture. So you can look at this change over time, look at different types of roots over time. Dynamics are really what we want to understand with the plants, how they're responding to environmental conditions. In this case, we've taken an image of these maize roots and this large primary root that goes down to the bottom of the chamber, out of the view down here. We injected a pulse of water right at the bottom of the chamber. And we took another image, and then we subtracted the two images. And what we can see is the difference in the images is basically where there's been a change in the water content. And you can see that there was a strong rehydration of that primary root that was pulling water up into the system. We have some new root tips growing here over the time period between image 1 and image 2. We can see this area here of roots that didn't have access to that water. They're attached to a different plant out of the view here. And these roots actually were dehydrating. And so understanding how much roots can dehydrate and rehydrate under different conditions is something that's important for our modeling. One of the neat things that roots do is they explore and they grow out through the soil. And we can look at this little red circle here, this new root growing over time, over a 12-hour period here. And basically showing that as this root's growing, it's releasing water out into the soil around it. And it's hydrating the soil around it through release of various organic compounds, through release of water. And so this is altering the soil rhizosphere. It's changing the soil chemistry and creating this very unique interactive zone between the root and the bulk soil. Another thing we can do is track water movement through the system. In this case, we've got this is Holly, one of our student interns, that was injecting some water near some targeted roots. And in this case, what we've done is grown some maize seedlings, and you can see we can see the entire root systems of these maize seedlings. And then about 7 centimeters below this bottom root here is where we injected, in this case, deuterium, which provides a nice contrast to the hydrogen in the system. And we can track that uptake by individual roots, redistribution through the root system, rehydration of soil even around specific roots as water might be released into the soil, this hydraulic redistribution. And then movement up through the plant. And this is my rendition of a movie showing this. What's interesting is this initial sort of burst of vapor is what I believe is moving through as soon as it was a fairly dry system. And as soon as we put that first injection in, the vapor gradient was moving through the soil, followed then by the soil water, unsaturated and saturated flow, and then uptake into the plants. You can see the new root tips growing over that period of time. Here's another experiment we did showing a pulse of deuterium added to the surface of the soil. And what happens is the water largely comes down one side of the soil column and then is taken up by the roots. And you can see the roots revealing themselves over time. Some of the roots over in this zone are rehydrating based on water moving from the main roots out into the finer roots, and even rehydrating the soil around the root tips. We've also been doing some tomography, and this is a CT reconstruction of a maize seedling. And this allows us to really understand where the roots are in 3 dimensions. Because it's difficult to really know when you're looking at just a 2D images, which roots are overlapping, which roots are touching, where they end up in the soil. And by having a 3D understanding of the root systems, then we can stop at some point and know where individual roots are, and they're isolated from other roots. And then we can do some dynamics over time. In this case, we were looking at water uptake by individual roots following a turning on of a lamp. And what we see here is these different roots shown by the yellow and the green and the red are responding differently to the water availability. Some of them take it up right away. This red root doesn't receive its water as quickly. When the lamp is turned off, water content and uptake declines in some of these roots. Whereas in the stem is basically accumulating that water over time at a greater rate during when the lamp was on than off. And so another way to look at the dynamics and function of individual roots. Some new work we've been doing is really exciting, is trying to do fast tomography and using machine learning to reconstruct these 3D images in high resolution. And as we start getting into these higher resolution dynamics, what we're getting into is much greater uptake of or greater generation of data, which requires some new understanding of how to process that data in real time. That's something we're really hoping to move forward with artificial intelligence, with larger computers hooked into the beamlines that can then process these data in real time. Here's Venkat, who's the image reconstruction scientist, doing an injection on this maize plant. Here we've got different types of soil that we're interested in looking at water movement and 3D reconstruction of roots within different types of media. You can see what that looks like. Much greater water retention and darker pixels here for the soil with more clay. The soil with just sand is almost completely clear, transparent. And here's sort of a 3D reconstruction of some of those roots. But this was some data we're just now analyzing, and we just collected in a couple months ago. And we were having trouble with the server crashing because we were filling it up too fast, and it was having trouble transferring it because we're generating so much data over short periods of time. And so this is a challenge that I think we'll all have going forward as we get better detectors and better beam lines, is how to deal with this data as we move forward. These were 0.1 second exposures. This is sort of what we're looking at through here, through time, and you can see this. He's identified a root here and doing these really fast reconstructions. What we're seeing then is that the deuterium pulse we added to the base of this system is then displacing the water in the system, and the attenuation signal is declining. And so these are just really raw data, but basically this is showing that the root is taking up deuterium and displacing the water in that system. If we look at a non-root zone with just soil, we see much slower movement of the deuterium through the system. And so we've got a lot of data we're processing from this. We really want to look at and understand how fine a resolution roots we can measure, and how fine a resolution rhizosphere around those roots we can measure. And looking at those dynamics. Soil water retention curves are a key component to understanding ecosystem function. Basically, these water retention curves describe the amount of water that's released under different degrees of drying. And it's based on things like soil organic matter and soil texture and soil bulk density. And these are often just estimated based on sort of bulk equations. When in fact, we can see that there's very different water release from different positions within the soil due to the spatial variability of the soil itself. And so what we can do is we can do this offline or online. We can sort of create these water release curves. This is showing a water release curve from the beam line. And that gives us these different parameters describing water release, residual water content, saturated water content, and some of these parameters that describe the release of the water. And these are needed for the Earth system models to project the amount of water that's available for plants, in which they can then either continue functioning or begin to shut down and quit taking carbon out of the atmosphere. One of the things we found with looking at this bottom left figure is looking at water release over time, applying a model based on those sort of known hydraulic parameters to our rooted plant system. The data are shown here. Soil water content over time are the dots, the blue dots. And when we apply our known water release curve, we measured on just plain sand, we get this black line. And so this black line is what we're actually, the soil water is what we're actually modeling, when in fact, in reality, with the plant and the root's presence, we have a much different response. And so the water release curve is incorrect. that we're using for the model. And we had to change this parameter a couple orders of magnitude in order to match, using the orange line here, match the water release. And so that led to sort of a new line of research focused on, well, just how much and how are roots and mycorrhizae affecting the soil water release parameters. This is showing actually water uptake through time versus root diameter. And this is something that's really interesting and needed, showing that the finest and the highest, smallest diameter roots are taking up the most water. And so we're actually able to quantify this water uptake rate versus root diameter. And thus, if we know our root diameter distribution through the soil, we can have some understanding of water uptake rates that we can put into the model. So following up on that last slide, how are roots and mycorrhizae affecting soil properties? We've done both paired lab studies as well as studies using neutrons, and we see some really big differences. This is showing just porosity itself, this factor on the y-axis, declining dramatically from a regular soil without roots to one with just mycorrhizae, to one with just roots, to one that's fertilized, and then to one that's mycorrhizae plus roots. And so we'll be using these data to apply new parameters to the water release curves in our models. So this is really exciting. We're hoping to continue along this line of work. One of the challenges going forward and ongoing and going forward is the image processing, making it easy to do, making it fast, and making it user-friendly. And so a number of different types of algorithms are used by different organizations and groups. Here's the one that we worked on, a student worked on with us, was basically taking these multiple panels, multiple radiographs, and stitching them together, creating a mask where the roots are, and then basically assigning specific roots to the closest soil particles around it. And in that way, we're assigning, if we look at a change in the water content of this soil sort of ped on here, we associate that change with this root nearest to it. And so here's an example of normalizing one of the images. In this case, we had a lot of focus on this main root system out here, segmenting the root, figuring out what the thickness of the root is with the software, mapping the root through this distance mapping to the closest soil particles. And then also, we get our root diameter distribution and understand where it is associated with the soil. And based on that, then we can look at root diameter versus water content, both in the rhizosphere near the soil, as well as further out into the bulk soil. And similar to the water uptake rates, we see a difference in the water retention around the smaller roots versus the larger roots that are more developed and suberized and not as active. And so these rhizosphere modifications around the smaller roots result in higher hydration despite greater extraction rates. And so there's another important component for modeling. So thinking sort of forward about what we're interested in doing here is, continuing along this line of research, but also thinking more about multiple scales, being able to look at this scale in the center bottom here, and then zoom in on an individual root, hair, or mycorrhizae and its interaction within the soil. Potentially leveraging different multimodal techniques, doing X-ray imaging as well as neutron imaging, as well as potentially using other techniques to look at some of these dynamics. And water's the easy thing. We want to move into, can we look at carbon? Can we look at carbon interactions within the soil? Can we look at carbon sequestration? Can we look at root carbon release against that water background? Or nutrients, nutrient cycling, nutrient uptake. Here's some of the technique, neutron microscopy. That's using these different gadolinium masks that filter and sort of align neutrons, basically taking us from fairly poor resolution in this upper right image to different masks that then take us down to much higher resolution. So that's one sort of technique we can use. Another thing we can do is using contrast agents. In this case, this leaf was placed into gadolinium solution. Gadolinium has a high cross section for neutrons. And we can see that gadolinium progresses up the vascular system into the progressively smaller and smaller veins. And so that's another way to look at the dynamics of function in situ of nutrient movement. Just briefly, some of the things we'll hope to do in the future is we're going to have this Hypha beamline shutting down, and hopefully simultaneously this Venus beamline will come up. That'll be at the spallation neutron source, and that's where we'll have our energy selective imaging. That will be really useful for working with plants and soils. And there's a proposed new beamline for a second target station that will be really useful as well. That'll allow some more advanced, higher resolution neutron grading interferometry that hopefully can help us look at things more than water, looking at more of the rhizosphere structure and hopefully at nutrients and carbon. One of the key things as we move forward and try to integrate work across groups and equipment is multimodal experiments, really trying to plan out how we can leverage different techniques to provide complementary information. And so the future of sort of where we're headed with this is continuing to look at these root and mycorrhizal interactions, this rhizosphere dynamic, which has a really unique bacterial composition. Again, carbon and nutrients, a big target, soil physical and chemical interactions, water flow through various types of porous media. This could include, looking at CO2 injection into the soils. Carbon sequestration is becoming a hotter and hotter topic as we go forward. How can we apply knowledge to understanding how soils develop, how they begin to sequester and store carbon? Certainly below ground competition, that's important for the modeling, and understanding plant stress under drought stress or heat stress, particularly as we move forward into this new world. Thanks, and that's all the slides I had, and love to have any questions.
Thank you, Jeff, for a very nice overview of the work you are doing. We have a few questions in the chat. Maybe Nicolas, do you want to start?
I apologize. I logged in with my mobile phone, so right now I'm in a tram. So I believe the question I wrote was, how do the Hypha, you mentioned Hypha and the mycorrhizae, how do those actually help the uptake of nutrients and the water? And how do they help the plant, specifically the Hypha?
Yeah, that's a great question. Those are, it's a really critical biological interaction that most plants have with fungi. These are symbiotic fungi that either sort of grow around the root, or actually some of them will grow directly into the cells of the root. And they've evolved over time so that the root doesn't try to defend itself against that sort of incursion into its cellular area. And these hyphae are much smaller diameter than roots, and so they can access smaller soil pores. They can also, through release of exudates, they can change the structure and essentially mine the surface of soils to derive nutrients, particularly phosphorus, which is one of the key limiting nutrients in tropical soils as well as other soils. But that's a key compound that they are able to uptake at much greater rates than regular roots. And then these are then transported fairly passively through water moving through the hyphae or through the through the rhizomorphs and towards the root system itself. And the fungi in exchange get some carbon that is exudated out by the root. It's a symbiotic relationship.
Thank you.
Thanks. I think Manuel also had a question.
Yeah. So first of all, thanks for the talk. It was very interesting. My question is about quantification. How do you go about with quantification? Because, from my experience, quantifying water with neutron imaging is quite complicated. Do you have some kind of calibration samples that you measure? Or you do measure the background due to hydrogen and then subtract it or correct for it?
Yeah, what we've done is use calibration standards, and so we've put in different thicknesses of water into our beamline. We've also done that with our hanging water columns, where we have known amounts of water at different positions. But the original work that we're basing our quantification on is several papers that were put out based on calibrations at the beamline using the same soils and root-free soils. And that is a challenge, and it's especially a big challenge when you start thinking about leveraging deuterium, D2O, because that D2O has a different cross-section, and we don't know quite how much it's mixing with the water signal. And again, as a dynamic system, there's a constant movement of water with the plants out of the picture. And so as soon as we start mixing the deuterium and the water, then that becomes more of an issue for quantification.
Thanks.
Any more questions? I see Björn has raised a question.
Yes, just a quick thought. Have you been able to determine if certain fungi contribute more to specific plant types with this approach?
That's a really active and a growing area of research at our lab, mainly not using neutrons, but using genetics. And there's a strong affinity for certain fungi with specific plants, and specific fungi confer much different and greater benefits to specific plants than others. And what, you know, it's our bioenergy program is pairing together different genotypes of poplar or switchgrass with different microbes that are found in the environment, different bacteria and different fungi, and then looking at the results. One thing that they're not able to really do is look at dynamics in situ. Rather, they see, well, this plant grew better. And when we extract it from the soil and measure it, we see that its root system is a little bit different. But there's no understanding of the dynamics of that functionality and just how that specific relationship creates a better result. And so that's where I think we can, you know, as we look at improving our detectors and improving our dynamics over time, looking at that interaction and understanding, is that related to water? Is it related to nutrients? Is it related to maintaining the rhizosphere in maintaining contact with the rhizosphere and the root? So there's a lot of research that could be done there with that.
Any more questions? I see Sindra has one.
Yeah, I have a question about your background here. What's do you have these plants in soil or in sand? And if you have them in soil, how do you know what that actually contains?
Yeah, that's a great point. You know, much of the work we've done and has been done has been in sand because it's so easy to work with. It's so transparent. So as we move more into soils, which we're trying to do now, we need to have an understanding of the soil physical and chemical properties. One of the challenges is as you move into more soils, particularly organic soils, you increase the background, you reduce the resolution, you increase the scattering and the attenuation of the beam by soil organic matter and by hydrogen that's attached to soil particles. And so it definitely becomes a much greater challenge. So those experiments have to be set up, understanding the limitations and really thinking about, you know, just what you're able to see.
Because it's my understanding is that soil is not just sort of a dead but complex mass, but there, I mean, there could be fungi there already, right? Like an ecosystem of other living species.
Right, right. And what we've done initially is start with a sterile system and add our plants and then add our fungi. And so we put in the fungi, the specific fungi of interest, and grow those out. And so in that case, we're able to look at, you know, these single interactions between a root system and that one species of fungi. It gets much more complex if you're using, I mean, certainly if you're using soil out of the environment, you're going to have hundreds or thousands of different fungi and bacteria in there. And so that's another question, I guess. And so that's something that we have to be taken into account.
I have another question, if that's okay. You started out with discussing climate modeling, and I'm wondering how do you think this data you're producing now, how is that going to feed back into the model? What's your prediction or hypothesis? What's it going to give?
Yeah, great question. You know, we work with the modeling group, and they've got all these different parameters. And they've got root distribution, they've got photosynthesis, they've got carbon allocation, they've got nitrogen cycling, they've got all these different processes. And what they've done is they've done uncertainty quantification, basically testing different parameter ranges for each one of these different processes and saying, well, you know, if we had a better understanding of this process, that would reduce overall model uncertainty by 0.5% or 1%. And so one of the things that sort of lights up on those sensitivity analysis are some of the root processes, carbon allocation to roots, other root dynamics. And so understanding how roots are functioning and particularly how they function during a dry period. That's another uncertainty that's currently using just a single parameter in the model that we're using. That says, okay, as the soil dries, root water uptake reduces along this trajectory. When in case that can vary very dramatically. And so understanding how that it's called the beta function, understanding how that beta function actually works during drought for different soil types, for different plant types, will allow us to refine that parameter and reduce the uncertainty in the overall model.
Thanks.
Yeah, these models are really incredibly complex, and they've got, you know, they have different species, they have different plant functional types, they've got different biomes, and so there's so much information out there that is just sort of bulk information that's used. And when you start drilling down into, well, what do we really know about, you know, this plant system? We can be, there's a lot of improvements to be made in specific parameters applied to specific plant functional types.
Thanks. One can only imagine the sort of computational machinery that is needed to model such a complex system.
Yeah, these are great, and you know, it's nice that if we look at the AR5 report from the IPCC, the model variability 5 years ago was greater, and now we've shrunk it a little bit over these last 5 years just by improving all these different parameters and representations over with the new generation of the models.
I see we also have a question by Emma in the chat. Please, Emma. Did we lose Emma?
I see that about mycorrhizae in the forest.
Okay.
So, and depending upon how the forest is harvested, it can take a long time for species.
All right, now I'm here. Yes, I heard that if you harvest the forest, like in the way that is done in most places, with large areas cut at once, it takes time for mycorrhizae to re-establish. So the spores blow in from the surrounding, but it can take a long time. And that the forest mycorrhizae make up a very large content of soil organic matter. If it was like, yeah, it was large. And it made me think whether the annually cultivated soils are deprived of mycorrhizae. Do you know anything about it?
Yeah, I think that's an important issue, and it can range from initial colonization of a denuded landscape with mycorrhizae to more developed systems. I think one comment on that would be that a lot of seedlings that are sold have mycorrhizae attached to the root systems. And so they've been grown and propagated with beneficial mycorrhizae, but then when they are planted out in the forest, they have those mycorrhizae with them already. And that provides a benefit for them. Areas that have had volcanic activity or had landslides where the topsoil is completely gone and all the mycorrhizae are gone. When people went out and started trying to plant in those areas with just regular seedlings, the seedlings had a very difficult time and many of them died. And that's when they started realizing that they needed to put the mycorrhizae on the roots, and then they could get really strong seedlings growing in those nuded areas.
I was thinking when I heard that on the radio about the forest mycorrhizae, that maybe it would be really good for us to have like lines of perennial species in the agricultural land to make sure there is always a mycorrhizal amount in the soil also. Now I will stop.
Yeah, no, it's really interesting. I think mycorrhizae is amazing. We were in a system in Ponderosa Pine in Oregon, and we were sampling mycorrhizae, and it's just basically pure Ponderosa Pine. And we collected a soil sample next to a tree, and we took it back and we found 15 different types of mycorrhizae. And we took another core a meter away, and we took it back and we found another 15, but 13 of them were unique species. And so the degree of diversity out there is really amazing. And we don't really know which species are most important, and that importance may change as environmental conditions change. It's a great topic of interest.
Thank you.
Thank you. And Jeff, this will keep you busy for quite some while, this diversity. Any more questions? If not, then let's thank Jeff for a great talk. I hope we can lure you to Lund for some experiments in a couple of years from now.
Absolutely, I'd love to come over, especially when Hypha goes down. We'll be down, and I'll be looking for experiments to do. And thinking, Kim, with the work that you do, how we might pair some measurements at ESS with the Max 4. These goals across scales.
Looking forward to it. I think this is
thank you all.
Great way to end this talk. So thanks, Jeff, and thank you to the audience for good questions.
Thank you.
Thank you.